Hi, my name is Jennifer Waters, and I'm a PhD student in Carrie House's lab at San Diego State University. And today I will be giving you a presentation that goes over my work in ovarian cancer called A Dip into Ovarian Cancer, studying the role of pre-adipocytes in disease progression. So ovarian cancer is the most lethal gynecologic malignancy in the United States. And doctors refer to it as a silent killer. And the reason it's earned this nickname is because it's really difficult to diagnose. And because of this, most patients aren't diagnosed until they reach stage 3 or stage 4 disease. And at this point, the cancer has undergone metastasis. Metastasis is the process by which cancer spreads to new organ systems. And unfortunately, in the case of ovarian cancer, by the time it metastasizes, the 5-year survival rate drops down to 25%. So what this means is that in patients that have experienced metastatic ovarian cancer, only 1 in 4 patients will survive 5 years past their diagnosis. So this really highlights a need for treatments that better account for some of the changes associated with metastatic ovarian cancer. So ovarian cancer spreads originally from the ovary via small spheroids that spread into the abdominal cavity. And these spheroids secrete factors into the tumor microenvironment that promote migration and adhesion to distant organ sites. And my project is specifically studying the role of adipocytes in these processes associated with metastasis. And the reason we care about adipocytes is because 80% of ovarian cancer patients with metastatic ovarian cancer present with metastasis to the omentum. And the omentum is a fatty tissue that lines the abdominal wall, and it's comprised primarily of these lipid-containing adipocytes. And these cells have been shown to promote ovarian cancer growth because the cancer cells are able to convert their lipids into energy. Oh no. And the omentum is also comprised of pre-adipocytes. And these are the precursor cells that give rise to mature adipocytes. They're considerably more fibroblastic in phenotype, and they're highly secretory. And we're interested in understanding the way that these cells contribute to the tumor microenvironment. And we've done this so far using 2 model systems. So in the first one, we employed a xenograft in mice where we injected small doses of ovarian cancer cells and determined that the smallest dose that we employed with 500 ovarian cancer cells was unable to form tumors in the mice unless they were co-injected with pre-adipocytes. And at this point, they were able to form tumors, which we isolated, and when we harvested the cells from those tumors, we found that the pre-adipocytes were required for sustained growth. So, in short, the pre-adipocytes are supporting a tumor initiation phenotype in vivo. And we wanted to investigate some of the signaling that's going on between these 2 cell types. So we performed an RNA sequencing study that used a co-culture system that contains both the pre-adipocytes and ovarian cancer cells, and discovered that when these cells are co-cultured together, they upregulate a program that promotes ECM remodeling. And specifically, they upregulate a gene called IGF-BP5. So going forward, my work will be focused around understanding the way that IGF-BP5 contributes to this pro-metastatic phenotype and investigating the way that it coordinates ECM remodeling in ovarian cancer. Thank you.